Okay, so good morning. Welcome to the Exam 2 review. Today I'm just going to cover a couple of key things to look at for Exam 2. I have a Jeopardy game, which depending on how much time we have, we'll play it, or maybe I'll just go through the questions and answers so you kind of see what Dr. Curley might ask on the exam. And like I did last time, I went through the lecture study guide, and I'll give you all key things to look at for this exam. Um, Thank you for your patience. I appreciate you all joining. I'm sorry I'm doing this on Monday, but this was the only day available I had before the exam. So I'll go ahead and get started. So we're going to cover very briefly blood vessels, hematology, lymph, immunity, and then any questions you have. And then, like I mentioned, we've got that Jeopardy game as well. First one for blood vessels, just recall the flow of blood vessels, how you go from arteries to arterioles to capillaries, then into venules and veins. Don't forget the concept of arterial branching. We like to have, why do our arteries not just stay as one big artery everywhere, and why do they have to branch out into different muscle groups, different tissues? And that's for perfusion, surface area, lower blood pressure, and allows the movement of fluids and solutes, things like electrolytes, that kind of stuff. And the three types of capillaries that we have the continuous, which is in most tissues. We have fenestrated, the ones that have little holes. Those are usually in the kidneys and small intestine. Can everyone else hear me, or is it just Arnisha that can't hear me? Looks like I see you're connecting to audio, Arnisha. So I wonder if it's on your end. Let me throw- uh, Arnisha, can you hear me now? Okay, perfect. Awesome. Thanks everyone for chatting in on that one. Like I said, going back to the capillaries, continuous are available in most tissues. Fenestrated, those have tiny holes in the tissue in the capillaries. Those are usually found in your kidneys and small intestine. And then discontinuous or sinusoids are usually found in liver, bone marrow, and spleen. That's where the artery will end and just have a big open hole at the end. Hematology. Okay, so the biggest things to remember about hematology. Uh, know what makes up blood. So remember, we have our formed elements, which include our red blood cells, our white blood cells, and our platelets or our thrombocytes. And then the processes they're all involved in. So red blood cells specifically are there to carry oxygen to our tissues and get rid of CO2 and other waste products. White blood cells, those are primarily involved in immunity. And then platelets or thrombocytes are usually involved in hemostasis or clotting to prevent blood loss. And then in plasma, know that we have water, mostly made up of water, but we also have proteins, primarily albumin, globulins, and fibrinogen. We have ions and nutrients and waste are in plasma. Ideally, you're able to identify what each of the big proteins do in the plasma. So what's the purpose of the albumin, the globulins, and the fibrinogen? And that can be found in your notes as well. So uh, also understanding, 55, 45% split between the formed elements and the plasma. So you know that we have typically more plasma than formed elements, that kind of thing. Is this not? There we go. Okay, so homeostasis. So we kind of discussed how all of our blood cells are coming off of one main stem cell. Um, Homeostasis and erythropoiesis. This is kind of in my way, but hopefully it's not in your way. Okay, there we go. Is the process by which we make red blood cells in the body. So why do we need more red blood cells? Usually it happens because we have low oxygen conditions or hypoxemia. Different things that can cause you to not have enough blood or really enough oxygen. The one that everyone thinks about is you have a hemorrhage or you're losing blood because you have a clot, you're bleeding out, that kind of thing. The two that people don't really think about is when you go to higher elevation, it's a lot harder for your red blood cells to transfer oxygen and gases. And so you need to have more red blood cells to make up for that. And then when you exercise, same thing. As you're running around and exercising on that first day, you're probably short of breath. You need more oxygen. And so the body will go through the process to make more red blood cells for you. So that way you exercise again. you have more oxygen available for your body and you're not as tired. I like to phrase this as, it takes about 3 to 5 days for you to get brand new red blood cells. So if you're working out on day one, you're exhausted, you're breathing hard. If you go work out again the second day, you're still going to be breathing just as hard as you were on day one. But if you wait a week, the following week, now your body has those fresh red blood cells in the body. And then when you start working out, you'll actually feel a little better. And it goes in reverse. So if you work out really heavily, you've got great red blood cell count. If you stop working out, your body says, okay, I'm wasting so many nutrients, so much of my energy making these red blood cells that I don't need. So it just starts getting rid of those red blood cells. And over time, you're going to go back to a homeostatic balance of, okay, I need this many red blood cells to do my regular day-to-day walking, the occasional exercise, whatever it might be. Your body doesn't like to waste anything. So red blood cells can go up or down based on your activity level, where you live, and obviously if you're bleeding out. So remember that is a negative feedback loop. So kind of like I mentioned, your body will make as many red blood cells as it needs, and it'll get rid of the red blood cells that you don't need. The other thing that people kind of mix together is homeostasis and hemostasis. So homeostasis is the production of new blood cells. Hemostasis is a way to keep the amount of blood cells that you have in place. So stasis means steady or frozen. So what your body's trying to do, if you have an excessive blood loss, is I need to be able to stop the blood loss so that I can at least keep the blood cells I have, and then I'll go back and ask for more red blood cells. Um, do you remember where the blood cells come from? So the kidney will release EPO or erythropoietin, and then that's what triggers the formation of those blood cells in your marrow. They go through different stages, kind of like you see here. You've got the pronormal blast, you have the normal blast. Um, I think we use different terms, and I got the notes here. So we'll start with the prourethroblast, and then the normal blast. Or sorry, the erythroblast, normal blast, and then the reticulocyte, and then the erythrocyte or red blood cell. What's happening in this process, and ideally you can describe this in a few keywords, might be a very good written question on this exam, is what happens at each stage of the red blood cell development, and how does the red blood cell at the very end look like this concave shape disc formation? So what ends up happening is you think about every other cell that we have, our cells have organelles, have like things like ribosomes, endoplasmic reticulum, nucleus, all those things. Those are built into the cells to help the cells regenerate, create what it needs, get rid of its waste products, that kind of thing. So the red blood cells, when they first start as a prourethroblast or erythroblasts, they still have those organelles. Those organelles are there to help the red blood cells start growing and developing, and then also to start making hemoglobin. And over time, as you go through this process, the red blood cell will start making hemoglobin and start getting rid of its organelles. So it starts getting rid of its endoplasmic reticulum. It starts getting rid of its ribosomes. It'll eventually get rid of its nucleus in the normal blast phase. And then once it's done that, the reticulocyte gets released into the blood cell, or sorry, into our bloodstream. And its reticulocyte is mostly a red blood cell with just a few little organelles left. And then over the next 24 hours, it gets rid of all of that stuff and it becomes the red blood cell. The reason the red blood cells can't last that long is it got rid of everything that it needs besides the ability to hold hemoglobin. So it can't repair itself, it can't reproduce. And so as it gets used up, it takes damage and it can't repair itself. And then over course of 3 months or so, it'll then be so worn out it can't do its function, and then it gets recycled, and then we make new red blood cells later. That is a very generic description of homeostasis, specifically, erythropoiesis. So be able to describe that in your own words and be able to kind of write it out, just what each phase is, and just give a brief description of what they do. That's something that I would recommend knowing for your exam. Hemostasis, the process of clotting, is another thing you want to be able to discuss, again, in very brief terms, be able to go in there and talk about, all right, I have a rupture in one of my arteries or veins. I'm losing blood. Now what happens with the platelets? How do they communicate with each other to cause more platelets to show up to then start creating the clot. What happens during the clotting process, and then immediately after the clotting process to stop the bleeding, and then what happens at the very end to then clean up all the blood clots so you don't have this large clot in your bloods in your veins or your vessels or your arteries. So being able to kind of walk through that really quickly is also a very good thing to know. I think that is on page, oh, I don't really have page numbers, but there's a whole hemostasis mechanism of controlling bleeding, which involves these three steps of vascular plasm, platelet plug, and then coagulation. Be able to kind of describe that very briefly. If you notice, I'm talking about a subject longer than some of the other ones. That just shows you that's probably a more important subject to look at for this exam versus like blood vessels, for example. And then if you have any questions during this, I didn't mention it, just throw it in the chat and I'll go back and answer those for you. And if you don't have any, that's cool. But you can either blurt them out or raise your hand or put them in the chat and I'll get to them. Lymphatic system. Lymphatic system is, if you kind of notice, it's pretty simple. There's really not too much to ask about lymphatic system, but there are going to be some key things to remember. One is the lymphatic flow. So recall that if you cut your body in like a square, the right side of your body, so half of the face, right side to your abdomen and across, that all goes to one lymphatic duct back into your blood vessels, into your vein, and then the other three-fourths of your body goes to the other side. So be able to describe if a question might ask you something like the lymph's trying to go back the to the blood vessels and it's coming from your left leg. What flow path do you expect to go from your left leg back into your blood vessels? And so you kind of show here how the right lymphatic dumps into the right subclavian, and then the left the thoracic duct dumps into the left subclavian vein. Again, that's a very easy question to get asked. Hey, tell me the flow path from the right leg, the left leg, the right arm, etc. And you kind of have to know which way. A very easy way to get confused is if we tell you, hey, tell me the lymph path from the right leg. You'll think, oh yeah, right lymphatic duct. Like, no, that's thoracic duct. That's a very common one that gets asked. Because you have to really understand where the flow is going. The other one is lymph nodes. Remember that the way lymph nodes are, they're kind of more of like, what's the term I'm thinking of? Like TSA, if you've ever flown before. So everyone kind of gets into the airport, they get into the lymph node, and then in the lymph node you have a bunch of immune cells, and they start screening everything. So they make sure that it's your cells or that the stuff getting through is nutrients, whatever it might be. And then if it sees somebody that's not supposed to be there, doesn't have a passport for you, if you will, and an illness or an infection, a bacteria, whatever, then the lymph node stops the flow and it gets a bunch of immune cells to go into there. That's kind of where you start seeing the swelling, is it cuts off the flow out of the lymph node so it can then target and destroy whatever illness or bacteria or infection is in there. And then it can feed that back to the rest of your immune system to then tell them, hey, I caught this at the TSA gate in let's say the armpit. Let's look around the rest of the body to see if we can find one. And then just remember your key organs for the lymphatic system. So you got your tonsils, you've got your thymus, which won't be available in adults, but you have your spleen is your biggest one, and all you also have like your bone marrow, and then obviously your lymph vessels, your lymph nodes, etc. Okay, so I briefly covered this on Tuesday, was the immune cells. Biggest thing to look at for this one is you need to be able to describe or identify which immune cells are going to be involved in what kind of infection. So I kind of threw it up here. Neutrophils, remember, those target primarily bacteria. Eosinophils are very good for parasites. Basophils, they are more involved in allergic reactions. They don't necessarily kill anything, but they'll release things like histamine and heparin, which are used to either cause swelling and inflammation, and it tells the body, hey, I need you to send more immune cells to this area. And it will also do like an anti-clotting. So some bacteria, some other viruses or parasites, they like to clot or cause clotting in the body. And so the basophils prevent that from happening. So it forces that bacteria out and that into the open so that the rest of the immune system can see it. And then understand that lymphocytes and monocytes, those are involved, or monocytes specifically, they'll become macrophages, and then macrophages will then go and clean up everything. They're very similar to neutrophils, where they also kill bacteria, fungi, etc. And then the big ones that we really want to for third line of defense or specific defense is the lymphocytes. So lymphocytes will become the B cells and the T cells. Understanding which of those cells will interact depending on the type of infection. So viruses and cancer, those typically are infections that happen inside of our cells. So the T cells are involved to kind of figure out which cells have a virus or an infection. I'll say virus and cancer specifically. And T cells are responsible for destroying our cells that are infected. B cells, anything that's outside of our cells, the B cells release antibodies. Antibodies will then clump around the bacteria or the fungi or the parasite. And then specifically, the antibodies will do a couple different things. They flavor the bacteria. They cause the bacteria and the antibodies to kind of clump together to make it easier for the immune system to see. Um, and then they're also involved in something that's called helps with complement proteins. So complement proteins are just like add-ons. So complements kind of help the immune system target other things and destroy some infections and that kind of thing. This is a very easy question. I'm pretty sure it's going to be multiple questions on your test. Again, depending on um, which type of infection she gives you, and then also what kind of cells you should be able to expect to be involved in an immune response. We did cover the first line defense, mucous membranes, skin, um, fever is part of it too. Be able to kind of describe the stages of if you get an infection, what first line barriers you should have, what second line cells are going to be involved, and then what third line or specific immune cells will be involved for your specific infection. This one's just a nice little summary of your innate versus adaptive immunity. So innate immunity is typically your first line. That's your mucous membranes, the skin, the acid in your gut, the alkalinity in your small intestine, the hairs in your nose, the saliva, things like that. Things that your body already naturally produces. It's just designed to slow or stop the infection before you have to start getting your immune system involved. Second line, that's when you start getting into, or the adaptive, that's more of where you start getting all your specific immune cells to try to target that specific type of infection. This is just an example of an innate versus an adaptive immunity. So things like inflammation, they kind of cause this like swelling effect, and then also creates like a bit of like a heat kind of thing in your body. So when you have like a fever, the heat actually enhances your immune system. So contrary to popular belief, you don't also necessarily always want to drop your fever below 100 degrees. You're actually making it harder for your immune system to function. And in turn, bacteria and other fungi, they like body temperature. And so by having your body go up to a fever, it actually slows down the growth of bacteria or fungi while also enhancing your own immune system. Typically, if you let your fever run for a day, sleep it off, cover up, whatever, your fever is usually gone the next day because it's already dealt with the infection. It's always when you have that prolonged fever for multiple days, that's when you have to start using medication to control it because the infection that you have is so strong, your body is trying really hard to kill it. And over time, excessive heat will also damage your proteins and then hurt your body. But that initial, first 24 hours, you start feeling that fever. I just bundle up, I get under the covers, I go to bed, and I let my body warm up to try to kill the infection. And then the following day, I'll take medication if I need to. But just in general, the innate immune system, right? The infection comes in, you have inflammation, you have dendritic cells, which are kind of a type of immune cell, or a type of macrophage. But you have macrophages, you have our generic um, immune cells, our second line cells. Those typically tend to attack during the innate phase. And then the adaptive is where once the body figures out whether it's a bacteria or a virus, then it can start sending T cells or B cells specifically to target that infection. And then it creates a memory. So the next time you get infected with something again, if it's exactly the same, your body remembers and attacks it very quickly. But if it's something that is brand new, your body takes a long time to then figure out how it's going to attack it and then goes on and attacks it. So a new virus, you'll get sick for a few days, maybe a day or two. Something that you've had before, like maybe even like a flu strain, give it 6 to 12 hours and your body's already kind of attacked it. Okay, so all I got for this one is, trust yourself. You do know more than you think you do. Um, does anyone have any questions right now before I roll into the Jeopardy game? And again, feel free to raise your hand, say it out loud, or type it in the chat.
Louise, I'm sorry. Can you break down the last class you kind of gave like a description of the immunity, like if we were infected with the flu, like
Hmm.
how like how it starts and how it ends.
Yeah,
I,
definitely. I'll kind of go back to that one. This is probably my best slide for this one. So remember the flu or any virus, viruses aren't living. They can't self-produce, they can't reproduce, they can't repair themselves. Viruses need to infect our cells and then take over our cell to then start producing brand new viruses. So that is done inside of our own cells. Bacteria or fungi or parasites, those can cause infections outside of their cells. Typically, the bacteria can squeeze in, attach itself to like our lining, and then cause an infection that way. To kind of I'll give you a quick example. Um, I'll use something different that I use in class. I'll still use the flu because that's a very common virus, or I could probably use COVID-19. It's very similar. And then I'll use, if you've ever heard of valley fever, that's done by a fungus, but you have very similar symptoms between a valley fever uh, and also like pneumonia and also the flu. You have that same cough and shortness of breath symptoms. So let's assume I'm the flu. So the flu is going to come in. You're going to breathe it in. Your body's probably going to detect that right away. To try to keep it out of your body, you're going to have the hairs or the cilia in your nose. They'll try to trap it or flick it away. If it goes in the mouth, you still have like your saliva. Saliva has some antibiotic properties, so it can kind of help neutralize, let's say, this virus. Um, your body will also be triggered to do things like coughing. So if I breathe it in through my mouth, my body's going to want to cough it out. By coughing it, you also create a lot of extra like saliva, and it tries to trap it, and you kind of spit it out. If it goes in through your nose, your body starts producing more and more mucus to try to trap the bacteria in the mucus, and hopefully you can sneeze it out. Or if it goes a little bit deeper in, the mucus will trap the flu, and then you have little micro, I won't keep using the word cilia, but there's a different word for it, little hairs on the lining of your nose on the inside, and it'll actually sweep it into your throat and force you to swallow it so that it gets into your stomach, and then the acid will maybe kill it. And if the acid doesn't kill it, because there are bacteria, there are viruses that like that acidic environment, it moves it over into the small intestine, which is now very alkaline. And then that's hopefully, if it likes acid, it's not going to like alkaline. Most organisms aren't like, I like hot and cold, or I like alkaline and acid. It's usually one or the other. And so we can put it into the small intestine and that'll kill it there. Ideally, that's where your infection ends, is that first line. Protection just keeps that infection from even touching your cells. But let's assume that that flu virus is very big, or you took a big whiff of it, and some of the flu viruses are able to attach to your surface cells. I'll call them surface cells. The way that the virus will attach, it'll kind of dissolve its lining, and then all of the viral parts go into your cell, and then it'll start taking over your cell and start mass producing. So if you remember that we have little flags on our cells, kind of like our blood cells do, the antigens. We have antigens on our cells that say, "Hey, I'm okay, I'm good." And if we ever have an illness or infection, it'll turn into a different antigen saying, "Hey, I'm infected, you got to get rid of me." So your body's constantly searching and going through and checking these antigens. So say it's like a hand, another hand comes up, checks it, okay, you're clean. I'm going to the next one, clean, clean. And then eventually you'll hit one that's, oh, you're infected. Usually these are done by the macrophages. They'll go in, they'll destroy our cell, take the chunks of the virus, and then bring it up to our third line cell. So the third line cell in this case would be the T cell or the helper T cell. The helper T cell um, will get this piece of the virus. It recognizes it or it doesn't recognize it, but it'll get a piece of it and it'll say, okay, hey, body, this is our suspect. We need to attack this specific chunk of the virus if you see it. Let's start looking at all of our cells to see where it could be located. The helper T cell, or we call it the general cell, they're the ones that then tell the rest of the immune cells, okay, I need B cells here or I need T cells here. For a viral infection, it's going to start telling the cells to turn into T cells. No, there we go. I got 10 minutes. So it'll start telling the body to turn into T cells. And then the T cells, you'll have cytotoxic T cells, which go and actually go and do the destroying and the hunting. You'll have memory T cells that get produced, and those kind of stay in reserve and like in the background just to remember the infection the next time it comes. And then you have T cells that are involved in the infection after the fact. So they turn off the rest of the T cells because the one thing you don't want to do is you don't want the body to continuously be searching for and attacking other cells. It does take a lot of energy from your body to have an immune response. That's why you feel tired, you feel sore, you feel sleepy, low energy. It's because your body's taking all the things that you would normally do, digestion, um, some of your brain processes, anything that wastes energy, your body says, you don't need to be thinking right now. You don't need to be moving. I need to attack this. I need to attack this infection, and I'm going to take that energy from you to do this. So that's the third line response. The T cells will then go hunt this virus, get rid of it, and then the other T cell, I forgot the name of the T cell, but you can look up in your notes. It'll then tell that cytotoxic T cell, okay, infection's done. Good job, guys. We won. We're done with this. Your body will then get rid of those T cells, but it'll always have the memory T cells alive. Those will always exist. Those will always reproduce. And as long as you've been infected with something once, your body will remember that. Um, on the flip side, let's say this is the fungus that causes valley fever. Very similar symptoms. The fungus is going to come in, your body's going to try to sneeze it out, cough it out. It's going to try to use its hairs to trap it, mucus to grab it, swallow it, put it towards the intestines or the small intestine or the intestines or the stomach. What it's going to do differently is this fungus for valley fever likes to settle in our lungs. So it'll actually go into the lungs and it'll start settling on the edges of our lungs, the inside lining of our lungs. Our body's going to have a very similar response on the first line side, the coughing and things like that. On the second line side, or the non-specific immunity, that's when these innate immune cells kind of come into play. So you're going to have, if you remember, for fungal infections, I believe we said fungus are neutrophils. So neutrophils do bacteria and fungus. So the neutrophils will then go out and start trying to attack this fungus. And so will the macrophages. So the macrophages and neutrophils are typically involved in most responses. They'll try to destroy the fungus from the outside. It's not in our cells anymore. They can see it. Neutrophils can kind of blast it with these little bullet kind of things to kind of rupture the fungus. And then the macrophages can then go and they can start eating it up. They'll destroy a fungus. They'll bring that same piece of the fungus to the CD4 helper T cell. And the T cell takes that, says, okay, this is what we're looking for, tells the body that this is the infection we're trying to go after. But because it's a fungus and they can see it on the outside, instead of telling the body to make T cells, it tells it to make B cells. And the B cells will then reproduce into or the T cells or the B cells will then go out and then they'll make different antibodies that will specifically target that fungus. So then those B cells get sent to the scene of your lungs, releases antibodies. The antibodies typically tend to attach to the outside of, in this case, a fungus. So imagine like this is your antibody. If it's perfectly with this fungus, it'll attach to it on the side, and then other antibodies will attach around it. And by doing so, it keeps the fungus from clamping onto the linings of our lungs to then cause the infection. Let me see the specific terms for this one. I want to make sure I use the right uh, words for this one. I know I'm running out of time, so B cells. Okay, so there's four different things there are different ways that the B cells actually kill the fungus in this case. So it does neutralization. So that's where it clumps on the edges. It prevents that fungus from actually infecting the rest of our cells. Then it does agglutinization. So antibodies can attach to the fungus, and then the backs of the antibodies can attach to other antibodies. And so it creates this clumping effect where you have this big blob of antibodies and fungi. And it just makes it a nice big chunk of stuff for our macrophages to immediately see and then start eating it all up. Opsonization is essentially like a chemical that the antibodies release, and it makes the macrophages like ultra aggressive. They're very hungry. It's like sugar, sprinkling sugar on something or salt or seasoning. It creates the makes the macrophages. They might want to eat something over here, but they really want to eat this glump of fungus and antibody stuff, and they just aggressively attack it, and that's just what destroys the fungus faster. And then we talk about complement fixation. So there's a antibody antigen relationship and different complement proteins. Um, I know she didn't want to get too specific to it, but complement proteins basically help enhance the opsonization, agglutinization, and some even will kind of create these little tiny holes to make it easier for the fungus to rupture or blow up, and then the macrophages can come eat it up. Does that kind of answer your question, Arnisha?
Oh yes, thank you.
Yep.
Thank you a
Again,
lot.
yep, that's very specific, but again, it's not so much the words I'm using, more so can you piece together an infection of some sort? Since I only have 3 minutes remaining, I won't go through the um, I won't go through the Jeopardy, but if you want the Jeopardy, just let me know. I'll send it to you. It's Dr. Curley gave it to me. But I do want to go over the things to look at in the study guide. So let's go to hematology section. For hematology section, things I would really look at, functions of blood. So number one, components of blood, number two. Number four, why do red blood cells have the shape that they have? Number five, what are the functions of the white blood cells? What are the different types of white blood cells? I guarantee you're going to have probably 4 or 5 questions on just white blood cells because we have so many of them. Number six, what are platelets and the process of the whole clotting effect or hemostasis? We are going to have some blood clotting questions. Hopefully, we kind of did this in lab. You understand those a little bit, but if you're not sure, just go ahead and refresh it. But just think about the generic ones. If you see that it's clotting on an anti-B or antibody, what kind of blood type do you have? That kind of thing. Um, Number nine, the process or the stages of red blood cell maturation. And I talked about, and 17 is very similar to the other one. So really 15 through 17 are just what platelets do and how do they clot up. So definitely know that one. So I said 15 through 17. And then for lymph immune system, number one, so different cells and structures and fluids found in the lymph system. Remember that in the lymph system is the only system that absorbs fat. or lipids. So make sure you see what structure absorbs that. Compare and contrast lymphatic capillaries and cardiovascular capillaries. She loves these contrast questions. And so I look at, what makes a blood vessel versus a lymph vessel, and how are they slightly different? How are they similar? Describe the differences between innate and adaptive immunity, number 5. We went over that in detail on Tuesday. We went over it today. Again, being able to identify when an innate response versus an adaptive response comes in, that's going to help you a lot on this test. A lot of questions can come from just those two topics. Um, Number 6, again, innate immunity. What are the different components of innate immunity? We discussed that today. Um, Number 11, adaptive immunity, right? So that's adaptive immunity. That's your B cells and your T cells, and where would each of them be involved, and what are the processes that T cells and cells go and destroy these cells. And then that's all involved in 13 through 15. That's kind of part of 11. What do B cells and T cells do? So I'm going to get kicked off in like 2 minutes, actually less than a minute. If anyone has any questions, feel free to reach out to me. I'm glad we went the entire time. Um, and then, yep, I'll send out the Jeopardy to everyone in the group and to the rest of the class. But again, if you have any questions, reach out. I don't know how long I got until they kick me out, so I'll stop the recording now. But good luck on the exam tomorrow. I'll see you in the morning tomorrow.